So we'll have a brief Kahoot just to get our attendance first. It's just going to be the color one again, probably.
Only that applies from this year.
Okay. We'll get this pulled up. And then...
Yesterday on FaceTime, she was like, "Am I doing this right?" It shows me Dr. Shear's lab like you did. Okay, let's tweak it out.
All right. All right, let's see, color poll. Sure. We'll get people signed in on this. Let me get this set up. All right, so while everybody's getting signed in on that, we are recovering from the Unit 3 exam. No, there's no curve on it, and there's not going to be a curve on it. And I would expect a similar exam situation, let's just put it that way, for the final. So be prepared for that. Moving forward, Unit 4 is a little bit more based on recall stuff. So for those of you who are better with recall, I think Unit 4 will be a little bit more friendly to you with that. But keep in mind that you will still have Unit 2 and Unit 3 questions on the final exam, right? And so I'm going to try to include more discussion about our content, and it's going to get meta, but I expect you guys to try to participate in that so we can start working through thinking critically about some stuff. I think that's a skill that lacks in a lot of students now, and it could be you have the skill in you and maybe we weren't using it, but we got to kick it into gear, because I mean, when you guys get into the nursing school, they're going to expect you guys to be able to critically think and apply and not just recall. I had a student like in my night class last night who like I handed out the scratch paper and we were already like everybody had their screens pulled up, we were on lockdown. She was like, "Can I just like write down everything that I remember on here?" Like brain dump is what we call it. And I allow that in my class, but they don't allow that in nursing school. Just FYI. So you have to have that knowledge ready to use in your brain and put it into critical thinking situations. And I don't want to send you guys on to nursing school or wherever you're going after this, vet tech or wherever it is, and you guys not have your ability to learn instead of just study and memorize. And so we've got to do a little bit more. We've only got a month left. And just to make it clear, if you were one of the people that was relying on study guide, in my class, the way that I see it is that is not your fault. You might have other professors that might treat it as you should have known better and you shouldn't have been doing this. That's not how I see it. That was a tool that you got from the university, and the university gave it to you. That's not on you. Not. And if you don't know how to learn because all of your other classes relied on recall of terms and word association and things like that, because other classes, it seems, did rely on stuff like that, that's not your fault either. But I can't fix that in a semester, and I can't fix it in a month for sure. So it's going to be work. Learning instead of studying and recall, learning is hard. It's hard, and it is work. And I mean work. I don't mean like time consuming. I mean work. And look, this is coming from somebody that, yeah, I went through graduate school, but that's learning. And that's learning on a level that I have to be able to explain it to other people. All of this content and everything like that. And so that was a lot of work to get me to here. And I'm just asking you guys to learn the basics and being able to apply it in basic scenarios. And when you get into nursing school, you're going to have scenarios that are more fine-tuned to a patient situation, that all the other answers are going to be right, and you have to pick the most right one for that situation. And that's critical thinking. And I don't want you guys to get to that point and not be able to succeed. So we're going to try to do what we can. So today, when we get into chapter 19, if we have time at the end of chapter 19, we're going to discuss. And I mean, it's going to get meta, and it's going to happen at big levels. So we need to do that, and it's going to be hard at first. But I need to start getting you guys thinking beyond recalling what I wrote or remembering what my picture was, or like this word associated with this word. There were a lot of people who were picking, I can see it on the exams when I look at them, picking answers because these words were in this definition, but in this context, it doesn't mean the same thing. And there was a lot of that going on the exam. You know, and if you want to go over your particular exam, I'll be happy to do that for you, but you have to come see me. I'm not opening up the exams, just to reiterate. So if you want to come see me at your lab or in my office hours or when I'm at the SSOC, you can come see me and I'll be happy to discuss those things with you guys. Otherwise, I will be happy as I'm doing this, working through trying to really push critical thinking on my side of things. If you want help with that sort of thing in any of your other classes that you might be in, including anatomy or physiology, I will help you with that thinking in those fields. I'm not an expert in those fields, but if I'm sitting there like, hey, let's figure this out together and we can talk it out together, that might be more of a benefit to you than just flashcards or a Quizlet or something. So if you want to come see me and try to work through those things on any of your other classes that are STEM related, I'll do my best to help you wherever I can. And that continues, that offer will always be on the table even when you leave my class. You can always email me and just be like, hey, I'm having a hard time with, you know, learning how to apply these things for this class. Do you have some time? And I will always be there to help you guys with that stuff if you're having trouble with it. When you get to learning a lot, when you get to the nursing program, because many of you guys are going into that program, what you'll find is that the professors and the advisors in the nursing program are a lot less forgiving than me. They're going to expect you to do the work. They're not going to be as like, okay, let's sit down and discuss this. They're going to be like, no, this is your responsibility. And so if you feel like you're getting lost in that, I don't want you to have to feel that way. And that if you want help with learning that stuff, you can work with a partner, of course, but that if you feel like you want me to help with you when you get to that point, I will be happy to do that. Okay, so let's do our quiz for the day, which is just, what's your favorite color again? You can pick a different one if you want to. I don't care. This is just so I can get attendance. And then we're going to get into chapter 9. We're going to get into the diseases. I'm a person that I favor blues and greens, and I definitely favor like a teal. That's my favorite. But it's not my choice. I would pick blue if I had to choose blue or green. That would be me. Generally speaking. All right. Let's get into gear with everything. Chapter 19. All right, so we're going to get into the skin and eye diseases. I'm not really going to test you on the anatomy of skin. That's not really what I'm going to focus on my class, but we do know that we have the epidermis, which is your outer layer of skin, and then it's the keratinized, keratin. We say keratinized is the term for what had happened to it because of the keratin. That layer of, it's got a lot of dead skin cells. We've got some living skin cells underneath the dead skin cells, but it's created a barrier essentially, and those keratinized skin cells there kind of have a waterproofing effect from the keratin that is in them. Then underneath that, we have the dermis, which is where we have a lot of our glands and our like main follicles where the hairs will be coming out of and that sort of stuff, as well as the capillary network and then layer of fat and other cells that are supporting that tissue in the subcutaneous layer. Again, I'm not going to test you on any of that anatomy, but I will test you on keratin. And keratin is a protein that basically gives a waterproofing effect so that the skin can withstand damage and abrasions. Let's say that when we start talking about damage that is done to the skin from infection or from burning or any injury or whatever like that, that we'll be talking about some of the skin like effects, like blisters or pustules or vesicles, and like what the differences are in some of these things as well. A blister essentially is when you start having that separation between the dermis and the epidermis. So that keratinized outer layer separating from that more vasculated layer underneath in the dermis. Those things can start to separate, and we see that either from trauma or burns, but we can also see that happening in certain infection situations as well, which we'll get to today. I'm not going to talk about follicles or sweat glands, but if you're interested in the makeup of the skin, some of the aspects of the skin, like the keratin with the waterproofing, can help protect from infection. This includes sebum, which is going to be the oil produced by your sebaceous glands. So the oil on the skin is low pH and it's inhospitable to many organisms. There are organisms, of course, like Staph epidermidis that will be able to live in the presence of these oils and the lipids like the fatty acids that are going to inhibit microbial growth. Sweat is also low pH and it has high salt. And we talked about how gram positives have the thick cell wall that helps them survive the salt situation. Lysozyme is, we've also mentioned that previously, in our innate, it's one of our first line defenses. Our first line defenses are the defenses that are like naturally in place, like lysozyme, keratin, sweat, the acid in your stomach, these and eyelashes, nasal hairs. These things are first line defenses and they exist to prevent the microbes from getting in. Second line defenses was our innate system, and third line defense was our adaptive immune response. Lysozyme is part of that first line defense. This is an enzyme that breaks down peptidoglycan found in our sweat and our saliva. All right, the normal biota on our skin, again, they're going to take up the stadium seats that would be available on your skin and like not make it so that, you know, pathogens would be as able to grow there. So if you're using a lot of soaps that have antibacterial compounds in them, like chlorhexidine, you don't want to bathe with something like HibaCleanse every single day because you're killing off your microbiome and then could potentially be opening up seats for other pathogens to sit down on and cause problems later on. So our biota is there for a reason. They can survive the saltiness and the sebum. We have the highest concentration of our microbes and the folds in the warm, moist areas on the skin. The two most common microbes part of the biome is going to be Staphylococcus epidermidis, as well as Propionibacterium acnes, P. acnes, which you probably heard about in acne commercials and stuff like that. These guys are in very high concentration on the body. About 4% of the population might have Staph aureus. Not everybody has the same biome, right? Based on what you're exposed to in your life. And so about 4% of the population can have Staphylococcus aureus as part of their natural biome. So it's not like something that we'll say that is common for everybody to have. It isn't, but it's not, we would say not super uncommon either. So it's not rare, but it is definitely less common. And Staph aureus is a potential pathogen. Not all Staph aureus is MRSA, so I do want to clarify that. MRSA is methicillin-resistant Staph aureus, and so if it has become resistant to the antibiotic methicillin, then it's MRSA. All right, so these are some of the skin conditions that can result from infections. So a bulla is a blister, basically. It's a word for blister. A cyst is going to be an encapsulated lesion where we usually have pus collecting in it. A macule is where we have basically a lesion characterized by color change. We see this in freckle situations as well as tinea versicolor, where we have an actual like color change on the skin from the melanin production, something like that. Maculopapular is where we have color change as well as the raised issue of a papule. A papule is going to be elevated. It's a small bump that is elevated like a wart. It's not filled with pus or anything, but there is an elevation in the skin. So maculopapular is like in between these two, where you have a color change involved as well. Petechiae, we say it's small purpura. You don't know what that is. Petechiae is when you have your capillaries under the skin might have bursting blood vessels, and you have some blood leakage under there. And so petechiae are small leakages of the capillaries in the skin. Plaques are going to be flat, but they are visible lesions. Again, purpura, we have a reddish discoloration. We can have petechiae where we have kind of spidery breaking off, or purpura, or we could have larger areas that look sort of like bruises. Pustules, again, filled with pus. I think it says it in its name, but it's going to be a larger structure. Well, whereas cysts are mostly fleshy in nature, a pustule definitely will have always like a pus aspect to them, and it can be visible even from the surface. Scale, it's just flaky, and then vesicle is something that's filled with fluid. So on chickenpox, what you actually get are these itchy vesicles, and they're filled with fluid. Or when we talk about with herpes outbreaks, those are vesicles that will come along with the outbreak of herpes. So let's move on to our infections themselves. MRSA and other soft tissue infections. MRSA and other Staphylococcus aureus infections are going to be common causes of lesions. This could be in non-hospitalized people as well as hospitalized people. We see MRSA getting transmitted around in hospitals-like situations, but you can get MRSA and staph outside of the hospital as well, just from person-to-person interactions. If you get a boil that seems to have a boil or a pustule and it seems to have pus in it, but it's very localized, a lot of times that can be attributed, not always. A lot of times it can be attributed to staph infection. And those can be temporary, or later on, if you recover from it, can potentially be part of your biome after that. But here we have something that's hot to the touch. We have pus that's associated with it. This is usually introduced through breaks in the skin, typically, more often than not, or injury from shaving or something like that. So Staph aureus, these guys are gram-positive. They are cocci. They are non-motile. They are non-endospore forming. They withstand high salt, high and low pH. It says extremes in pH, but so it could be high or low, and they can survive within a large range. But we know that when we grow Staphylococcus aureus on mannitol salts agar, it turns mannitol salts agar yellow, and that's what this picture is showing us. So mannitol salts agar, we'll recall, has the sugar mannitol in it, and Staphylococcus is capable of fermenting it and creating an acid byproduct. They can be pretty resistant to drying as well. The other thing about Staph aureus is that it produces an enzyme called coagulase. And it does what it sounds like, it coagulates. What it coagulates specifically is plasma. And so we can mix your patient sample with some rabbit plasma, just some plasma from an animal, and in a tube, and you can buy this. And you can put your patient sample in plasma, and if it coagulates, your organism produces coagulase. And that would be meaning that you might have Staph aureus. Some other enzymes that it produces that are involved in its ability to cause disease might be hyaluronidase, where it digests hyaluronic acid, which is part of your extracellular matrix. The staphylokinase digests blood clots, DNase digests DNA. And so all of these enzymes are basically involved. These are things that can help it cause disease. And then these are called, there's a term for this, it's called virulence factor. We've learned about this previously. Anything that helps with the pathogenesis of an organism is called a virulence factor. So these are all of the virulence factors of Staph aureus. Again, we can mix your patient sample with some plasma, usually it's rabbit derived. And then if it coagulates it, we will consider it positive for the coagulase. And that's very indicative of that, especially if your patient already has a skin infection. Oh, I'm going to swab the skin infection and do this. So we don't do this with every single infection ever. But if we have an indication we think it might be staph, it's a good test for it. So here are some diseases that can be caused by staph and other organisms. This one's impetigo. Impetigo is usually caused by Streptococcus pyogenes or Staphylococcus aureus, or a mixture of the two. And what we usually see is that usually Streptococcus pyogenes will come in first, and then staph will take advantage of that situation and then overgrow. But it is very commonly associated with both of these. We have a flaky appearance and blistering of the skin. We see this commonly in children. We see that reddish sort of flaky skin infection. They can get this impetigo. So what happens is Streptococcus pyogenes usually comes in first, and then staph from the environment can come in and basically destroy strep and then take over like all the open stadium seats that it opened up for it. We have exotoxins that are produced by Staphylococcus aureus that can be exfoliative, which is why we see the flaking aspect associated with this, as well as that coagulase that will coagulate the blood and the plasma. We have Streptococcus pyogenes. So this is the other cause that can be involved in impetigo. So impetigo is defined by its symptoms, right? And so it can be caused by one or both of these bacteria. So Streptococcus pyogenes, which we'll learn about in our respiratory chapter in a little while, also associated with strep throat. So it can cause infections all throughout the body, just like kind of like how staph can do. But we'll hear a lot about Streptococcus pyogenes and Streptococcus pneumoniae throughout all of these chapters. They're going to pop up in a lot of areas. So Streptococcus pyogenes is a gram-positive coccus, and it is beta hemolytic, and it's very well known for being beta hemolytic. It is the cause of strep throat, scarlet fever. It can cause pneumonia, puerperal fever, necrotizing fasciitis. It's usually one of the more common ones that can cause necrotizing fasciitis. There's a lot of bacteria that can lead to necrotizing fasciitis. And if I'm saying this word and you're like, don't know what that is, flesh-eating bacteria. So this is whenever you get an infection in the dermis and it spreads within the dermis, and it spreads very quickly. So when you see like on shows, or if you've been working in hospitals and you've seen this happen, where they'll draw a line around an area of redness and then over time see how long it takes for the redness area to cross the line. When you have necrotizing fasciitis, that redness, that inflammation will cross that line very quickly. That's almost the indicator that you have necrotizing fasciitis. And if it's left to go for too long, the tissue that is affected will die very quickly. And so you have to debride that tissue. And so debridement is whenever you remove the infected and dead tissue called necrotized. So that's necrosis. So that's necrotizing fasciitis. This is one of the main causes of that. Bloodstream infections and rheumatic fever are also tied to Streptococcus pyogenes. So this guy puts his fingers in a lot of areas and can cause a lot of different diseases, not just impetigo. And it has adhesive elements that help it adhere within its host. And it also produces high hyaluronidase so that it can break down the extracellular matrix and get into deeper tissues. Again, it's beta hemolytic and can break down the red blood cells around its colonies. We have rapid tests that are available for it, and it is sensitive. We'll test its sensitivity to bacitracin. That can be basically us using Kirby-Bauer for an identification method. And so its size of the clearing, the zone of inhibition around bacitracin, can indicate that it is strep in addition to its beta hemolytic abilities. All right, next, that was, we talked about impetigo. We introduced our two culprits for many skin infections, which is going to be Staphylococcus aureus and Streptococcus pyogenes. Cellulitis. This is where we have infection in the dermis and the subcutaneous. It's a lower level infection. It's not just in the upper layers of the epidermis and the dermis. We could potentially infect the subcutaneous tissues. We have pain, swelling, and warmth, and usually redness of the tissue. And there is often not going to be any introduction through trauma. It's just migration of microbes through the upper layers of the skin into the deeper tissues, and then this is spreading of the tissues in the deeper tissues. We see this a lot in the lower leg, especially patients who have diabetes and decreased blood flow. We also have staphylococcal scalded skin syndrome, or SSSS. We see this in children and newborns. I had a nephew that had this and had to live in Kansas, and they had to drive him to Denver for special treatment because his case was so severe. This is a systemic form of impetigo. So instead of just getting the flaky, blistery area, like maybe in one area of the skin or on, the scalp or even on the face, like we're talking about, like systemic all over the body. So we have exfoliative toxins A and B. Remember, that's what was responsible for the flakiness of impetigo. It's what's going to lead to the blistering that we'll see with the scalded skin syndrome as well. So those exfoliative toxins are actually imparted by phages that infected the bacteria. Remember, we called that is lysogenic conversion. So lysogenic conversion is where a bacteriophage infected the staph at some point, and with that bacteriophage infection, the staph got these genes for toxins A and B. This is the case for a lot of our bacteria. A lot of our bacteria got their toxin genes from having been infected by a bacteriophage. So that's lysogenic conversion. See how it's coming around, guys. It's coming back. All right. So this will lead to the development of these blistery lesions, these bullous lesions from the exfoliative toxins that will lead to detachment of the layers of the skin and can lead to widespread desquamation. Desquamation is whenever you have skin cells detaching from the body. And so we will have these large blister lesions. This is on an adult, obviously, but where we have a arm where this was a whole blister area that was detached due to this Staphylococcus scalded skin syndrome in an adult. But on children, this can be happening all over their entire body. So these blisters that look a lot like burn blisters all over the body, just from staph infection, becoming systemic like that. All right, moving on to gas gangrene. Gas gangrene is also known as clostridial myonecrosis. Myo leading to like muscle tissue, and necrosis, death. So death of the muscle tissue caused by a clostridial species. So this is caused by Clostridium perfringens. So we should know automatically, if we're hearing Clostridium, we should know in our brains, we call it, oh yes, that was one of the endospore forming guys. And so Clostridium perfringens as a bacteria is found naturally in soil, naturally on people's skin, in the vaginal tract, in the intestine as well. It's very common bacteria. But when this bacteria gets into the deep tissues, remember Clostridium is an anaerobic bacteria. It's what we call an obligate anaerobe. And so it doesn't do well with exposure to oxygen. So in the presence of oxygen, it'll be an endospore. But once it gets into your deep tissue, it can start multiplying. And so in the deep tissues away from the oxygen is whenever it starts to grow and multiply. And that will lead to gas gangrene, because one of the things it produces during its growth process is gas, literally. So yeah, so this is an example of gas gangrene that is remaining kind of in this lower leg area of this individual. And this is what we call dry gas gangrene. There's also a wet version of gas gangrene, where we will see swelling of the tissues. We'll get blisters that are filled with black fluid from the necrotic release of chemicals. We can see swelling and filling of the compartments of the muscle from the gas that is being produced by the bacteria. And that leads to edema. We're trying to fight off bacteria. The fluid is flowing into the spaces, and then that fluid in the spaces will get released into the muscle tissue. And then these folks that can actually get gas gangrene, performing the wet version of gas gangrene, leaking out fluid out of like their bodies, out like, it rips their body open, is what we'll say. There's so much gas in there, it'll rip open the tissue. And so when we talk about things like fasciotomies, if you've ever heard of that, a fasciotomy is where somebody has an infection, we've got swelling, or if you have a compartment issue where the fluid can't, move freely from one area of the body to another. We will cut open their skin so that the tissue underneath can expand and swell and won't rip the skin, because it's better for you to be able to heal if we cut it cleanly than for it to rip itself open. And so it can get so swollen and filled with gas that it will actually rip people's skin open. And so this is something we associated pretty much back in the day when people used to fight with like bayonets and get like deep stab wounds from that. But we still see gangrene and deep tissue infections today. Like anytime that you have a deep tissue infection, this could be an issue. And myonecrosis is death of the muscle tissue. And this is a cross section of muscle tissue. And these white spaces that are in there are the gas that is being produced by the bacteria. And the bacteria are the dark spots on this. That's the Clostridium perfringens bacteria. These bacteria have several toxins that they produce, like collagenase and hyaluronidase, to get deeper into the tissue and break down the extracellular matrix, as well as other toxins that are going to lead to this inflammation aspect and tissue destruction. All right, moving on to our pustular rash diseases and vesicular rash diseases. So we call a lot of lesions pox. Whenever we see a vesicular rash, we might call that a pox. And so things are just generally categorized as a pox if we see these vesicular rashes. So that would be chicken pox, smallpox, and monkeypox. Monkeypox is similar to smallpox, but it's a lot milder than smallpox. It doesn't seem to be as contagious as our major form of smallpox. Also, hand, foot, and mouth disease has a vesicular rash that's associated with it. We mostly see hand, foot, and mouth disease associated with children. Chickenpox is caused by the virus. It's caused by a virus, okay? Chickenpox is caused by the virus varicella zoster. It is a herpes virus. Chickenpox is usually mild for most people. They'll get these itchy vesicular rash on them. And when you scratch them, it's the secondary infections that can cause, scarring. And so it's not the vesicular rash that causes the scars. It's scratching and getting a secondary infection, which is something apparently I was pretty bad about whenever I was 7 and I had the chickenpox. I have a lot of scars from chickenpox in my body. It can be life-threatening in people who have an immunocompromised situation, or when we have reactivation later in life and your immune system is weaker, when we see elderly adults getting reactivation of chickenpox in the form of shingles. Vaccines are available nowadays, and they are effective, and they can protect from the initial infection, but you can still get the infection later in life. And if later in life your immune system is weaker, it'll present more like shingles, just not in the same locations that we see shingles. And there's a reason for that, because shingles hides out in the nerves. And whenever you have an outbreak of secondary chickenpox, which we call shingles, it comes out on the nerve that it was hiding in. But if you're an elderly person and you've never had chickenpox and you get it as an elderly person, it'll present like shingles, but all over your body like chickenpox, but it'll be more severe like the shingles disease. Just because your immune system isn't as strong. Your immune system is the strongest when you're younger and a child. Okay, so I should be saying something. They're already getting the chickenpox, how common that can be. Incubation period is 10 to 20 days. So if you remember back to chapter 2, when we were learning about like the growth curve, about bacteria and all this stuff. Incubation period is basically a lag phase. So this is the amount of time that it takes for that infection to take hold and actually start having effects on your body. So this is a period of no symptoms. So from the moment you get chickenpox virus in your body, it takes 10 to 20 days for symptoms to start to appear. Remember that your first symptoms that you get in the disease is called the prodrome phase. And then once you get into the acute phase of the disease, you'll start having more severe symptoms. And those terms will come up in this unit a lot. So the illness, the lag phase, lag. So the lag period, the period before we start actually having the growth of the virus is just getting established and the immune system hasn't started producing to it. That's what we will refer to, that the lag phase and the growth period, or the incubation phase in the epidemic terms. Okay. So illness will last about 4 to 7 days, but we'll usually see lesions stopping about 5 days after, and most cases resolve completely and have completely healed up within 2 to 3 weeks. And some people get secondary infections from strep or staph. I definitely did. I'll be completely honest. I have scars from it. All right. So we have reactivation of chickenpox from, this is human herpes virus 3, aka varicella zoster virus, aka the chickenpox virus. Okay. So it's a herpes virus, human herpes virus 3. And people will have reactivation of this virus after the fact later on down the line when your immune system is weaker, or if you didn't have a very, just naturally, your reassortment of your genes didn't protect you very well from reactivation. So people can still see it younger as well. But that is called shingles when we have that reactivation. And we can give that the chickenpox vaccine to adults to make sure their immune response is staying strong for that protection. Smallpox is another vesicular rash disease. We have macular, then papular, then vesicular, and finally pustular. And so we don't just have only vesicular with this. It'll start out as basically like a skin lesion that's just darker in color, and then it'll eventually turn into these pus-filled vesicles as the disease progresses. There's 2 forms of smallpox, variola major and variola minor. Variola major obviously causes the more major disease. It's very virulent. It's extremely contagious. It can lead to toxemia, shock, and intravascular coagulation. Smallpox is a deadly disease. So we're talking about like anywhere from 30 to 50% mortality rate with some of these guys. So you don't just get the rash, but you get other symptoms as well that are associated with smallpox. Smallpox, before we move on to these other guys. Smallpox, the first vaccine ever invented was against smallpox, and we talked about this before. It was Edward Jenner, and Edward Jenner basically noticed that milkmaids weren't getting smallpox, and so they would get these pox on their hands from dealing with cows. That was cowpox, and that virus is called vaccinia. So vaccinia, vaca, cow, vaccinia virus. He used cowpox to inoculate his son, and then pushed his son out into the population that had smallpox, and his son did not get smallpox. So he was spared having to have this disfiguring smallpox disease and potentially dying from it because he had been vaccinated with the vaccinia virus. So vaccinia is still actually used today as like the gold standard for vaccinating against smallpox in countries that still vaccinate against smallpox. If you work in the military in the United States, the military still vaccinates against smallpox. Otherwise, the rest of the population of the US is not protected against smallpox. My mom likes to boast about how she has a scar on her arm from the smallpox vaccine. I'm protected. The protection only lasts 10 years, and they haven't vaccinated in well over. I didn't get vaccination. I'm 41 years old. So it was well before I was born that they stopped giving out the smallpox vaccination. So none of us in this room is protected from smallpox unless you were in the military. Were you in the military? Yeah. So
it's nasty.
It's not good. Yeah.
It's
So
gross.
they do what we call excoriation. They scratch into your arm and they want you to develop a nasty scab. And they say you're not actually protected until the scab like has fallen off and healed. So once you've healed, then you have the immunity, and it has to happen that way to get the proper immunity.
But it's not just like a little scab. It's
No,
like
it's big.
2
Yeah, it's a
inches,
big
like
scab.
this round.
Yeah, it'll be like
It's
huge,
gross.
and you are considered contagious with it. So you
And
could,
if I see
get
it
it to other
from the
people.
arm at the pool, I'm not getting it.
yeah,
Ever.
it's gross. So you don't want to have to. They used to do that, and like if you lived in other countries like Russia and Mexico, they vaccinate pretty much for all their citizens that have been born naturally to those countries. They'll be part of like their vaccine regimen and stuff like that. So you'll still see people get them in those countries. Yeah,
So it's just not common enough here anymore to warrant doing all that.
it's eradicated from the planet. Nobody has it. Yeah. So the reason they still vaccinate in other countries is for bioterrorism threat. Because, I mean, America and Russia and China all have frozen stores of smallpox in their freezers. We need to talk more about this, or is this pretty much self-explanatory? So yeah, it doesn't exist naturally.
Is that one tunnel in Sweden actually?
A tunnel in Sweden, what?
The tunnel in Sweden that has a whole bunch of like microbacteria and everything like that.
Oh,
It's
that's
just kind of-
100% real.
Yeah, so that exists, that's a thing, and so we consider that to be like our like vault of like living things. If we ever had like
It's
a
like the
nuclear
library
war
of
all over
nature.
the world, we could potentially repopulate the world with organisms again, the seed vault. And seeds and microorganisms, the seeds, there's a seed vault as well in addition to this, but like, there's all sorts of vaults like that over the world where they're like trying to like, hey, like we can't trust people. And we can't trust natural events either, right? But definitely not people.
It brought the werewolf back.
So.
Girl,
yeah, I heard
only
about
like
that.
a little, it's like 99% other wolf and then a little bit of dire.
Yeah, just a little bit, yeah. But anyway, so, but yeah, so this still exists in cold freezer storage. I can't actually verify that because I used to do research with the CDC and they told us about how they would, store their smallpox, and they have like specific things in place to protect people from knowing where it is. Like even they didn't know in which freezer it was, and it was constantly moved around. So they would move like, there's like, I don't know, a whole bunch of boxes, like 50 boxes that'll just get cycled around and nobody knows like which box, no one person knows enough about the information to know where it is at any given time. So if they had to do the data to like delineate it, they needed it for, I don't know why you would need it, but hey, I'm not the person, right? So, but that if they had to access it, they could, for those reasons. And I mean, we're not the only country that has frozen stores of it. There's Russia, there is China, and there's probably other countries that we just don't know they have stores of it. But naturally, in the population of all animals on the planet, this is not a natural infection anymore. It doesn't exist anymore. It's been eradicated from the planet since the 70s. So pretty cool. Vaccines have done that for us. But this used to be, I'm sure you guys know about the history of smallpox, right? It used to be something they used in biological warfare back in the day before they had like bombs and things. They would throw like blankets with smallpox virus on it into like the natives and be like, oh yes, we will come and help you, and then let the natives die off and then come and take over their lands and resources.
That's how Oklahoma.
Yeah, so, and they did that to Native Americans. They did that to the people of Mexico, the Aztecs and the Mayas. They did that all over the world. So FYI, that's a big part of human history. It was our first versions of biological warfare, that and the plague. So we'll get into plague later on. All right, so maculopapular rash diseases. These guys will have some darkening of the skin, but also have potential papules as well. So raised lesions in these guys. So measles, rubella, fifth disease, and roseola. We know about measles. We've heard about measles. So measles is like considered in our skin disease category, but it can lead to other effects besides that. Measles is also known as rubeola. Okay, not roseola. That's a different thing. Rubeola, and I won't test you on rubeola. I'll test you on measles. But it was used to be called that. We had a sharp increase in measles occurring prior to COVID-19 due to the anti-vax thing. And that this is from the textbook that was updated in 2022. I'm sure we could add plenty of new history to this textbook with what has just happened in this year regarding measles. We have, by the way, there are cases of measles in Oklahoma City now. We have patients at OU Children with measles. They're in the hospital. Let me just tell you that. They're not at home with the measles. They're in the hospital, y'all.
My cousin's currently in the hospital with measles. Her mom didn't.
They don't vaccinate.
So
Before the vaccine was introduced, in case you need more evidence, maybe a vaccine might cause harm, but which causes more harm, the vaccine or the illness?
the illness, people die.
That's the point of vaccination. So before the vaccine was introduced, measles killed 6 million people a year.
Mhm.
Educate your family members, educate your family,
I-
your friends, and your patients, please. Because 6 million people a year died from the measles. We talk about it being a skin rash disease. For some people it is, but many people will develop meningitis and things like that. Haley.
Okay, so my grandparents were born in 1982.
Uh-huh.
And they have like pre-existing health conditions and everything. They're COVID vaccinated like 4 times, whatever.
Right. The viruses with immunity.
They texted me and asked about measles, whatever. They would be considered like, oh yeah, you should probably.
They are at risk. They are at risk. Yeah.
What about like like my age group or like my kids?
So you will only know that if you get a titer for your antibodies against measles. If you're going into the nursing program, they require that you have an antibody titer. So if you're going to be doing clinicals in a hospital, they require as a hospital worker to have titers showing that your vaccines have been effective for you. So you can do that. They are not cheap. They're not cheap. And I don't think the nursing program pays for that either, right, Levi? I'm pretty sure like that you guys have to pay out of pocket to get your titers done or, yeah.
Just depends on where you go, like I had to pay $100 to an urgent care for the gold, whatever it's called.
Yeah,
test.
yeah, that's just the TB test. And so, yeah, you gotta consider like these tests and looking at your titer antibodies for working at certain hospitals and going into healthcare, you're gonna have to prove that you have these vaccines. Yes, there are exceptions if you have, religious background, but because you're a healthcare worker and you're interacting with people, they have the right not to hire you if you aren't vaccinated. So that is,
The booster, like my grandparents, she did the booster.
yes, get a
But, like,
booster. And if you get your titer, so to know on your own, you would have to go get a titer done and your kids would have to go get a titer done or whatever. But other than that, most people maintain a good antibody against measles. It's absolutely the most effective vaccine that's ever been developed in humanity. So it's a really effective vaccine and you will be protected for a long period of time. And most people don't need a booster against the MMR, but some people might. And according to, what actually would protect you, what antibody levels there are and what the hospitals require that you have. The hospitals require you have a higher titer than what like the CDC says would be indicative of protection. So FYI about that. But so you could go get that tested if you're concerned about it because it is going to be coming around. I don't know what my titers are, and so you could go get boosters. Boosters are going to cost you money too, right? So we love our insurance company, but they're not going to protect you from this stuff. But FYI, just so you know, so you're aware of this. 6 million people per year used to die before we had the measles vaccine. Go ask your grandparents what it was like before we had the measles vaccine. How many people would get the measles? Y'all ain't never seen the measles. So yeah.
But like as an average person with good health, do I need a booster?
Generally speaking, no,
I
right?
assume I got it at some point when I was younger.
Yes, most people got it when they were younger. You probably got it as part of your normal vaccine regimen if you were just following those rules. So most people will be protected and won't need a booster for that. Most people. There are some people who, like one of my students last year came to me and told me like my titers were low for MMR, so I had to go get a booster for MMR. And so, yeah, so be aware of that. Most people should be protected though. To achieve herd immunity, in order to protect folks like your grandparents, right? In order to protect those folks, we need 95% of the population to be vaccinated against MMR. And that's clearly falling. That's fallen below 95%. That's why we've got children that are in the hospital right now with measles. So vaccinate. The risk of death versus the risk of issues that you could potentially have with the vaccine, one of which is not autism. Okay. Moving on. So with the measles, we don't just have the rash associated with it. We have sore throat, dry cough, headache, conjunctivitis, lymphadenitis, fever. We have Koplik's spots. These are that's the rash that shows up. They'll start off as dark spots and then be raised spots. Those are called Koplik's spots. And those come later on in the infection. Small number of people will get laryngitis, bronchopneumonia, and secondary bacterial infections because they're fighting off the measles virus. And it's called the measles virus. That's what it's called. We can also have severe complications that can include subacute sclerosing panencephalitis. This is actual inflammation throughout all of the entire brain, and that can be deadly. So an encephalitis thing that can happen, we have meningitis that could potentially happen. This will lead to progressive degeneration of the cerebral cortex and can lead to death and coma.
So will you go back so I can finish writing the long one, just so I have the other one?
Sure. All right, so I think we've gotten the point across with the measles. You might just get a rash, and congratulations to you. When your neighbor's baby dies, who's that really on? Baby can't get vaccinated. Yeah. So some diseases lead to wart-like eruptions. HPV is what causes warts in people. All warts ever, genital warts, skin warts, whatever, are generally caused by some form of human papilloma virus. So a papilloma is another word for a wart. So there's 130 that we know of. There's probably more. at least 130 different human papillomaviruses. Only some of them are associated with genital diseases. We'll talk about HPV and cervical cancer in the last chapter, chapter 24, and sexually transmitted diseases. All right, so moving on to anthrax. Anthrax can come in a lot of different forms. It can be cutaneous, respiratory, GI, circulatory, but this is the cutaneous version. So cutaneous means that it's in the skin. So this is caused by a bacteria, Bacillus anthracis. So Bacillus anthracis is Bacillus species, so it is endospore forming. It's found naturally in soil, and it's very common in like livestock. And so people who have farms, they might have endospores from their livestock that can just be resting in the farms, and then they'll like cycle their land between livestock land and farming land. And when they till it, it'll cause the endospores to come back up out of the ground.
This happens a lot, and what you can do to prevent it is during season change, you can move them to over 3 acres away.
Yeah.
And then you till the land.
Yeah,
That's
so-
what we're currently doing with our
Right.
cattle.
So when we're saying that this happens a lot, like this is actually like farmers are well aware of this as an
Yeah.
issue. Like this is a common thing. This is not uncommon.
You
So
can
farmers are aware of this, and they have things that they'll do. They'll like not try to cycle right next to the land where they had the livestock, but cycle land that is 3 acres away or more to prevent, waking up all those endospores out of the ground. They can move into cuts or abrasions in the skin. And then these guys are not anaerobic. They don't need to be deep in the tissue. These guys are aerobic. And so they will grow in the skin. And this is an actual literal picture of somebody that has anthrax in the skin. So that black tissue there is what we call, I try every year to say eschar or something like that. I won't make you say it, but you do need to recognize that on the test. I think it's eschar. I think that's how it's pronounced. And these are whenever the endospores germinate in the skin, and that's leading to necrosis in the skin. It's a huge issue with bioterrorism because those endospores are easily spread and can be breathed in and then can take up in your lungs. And it's worse to have a respiratory version of this. So this is the least serious of all of the anthraxes that you can get. It still has a 20% mortality rate if it is untreated. It's a huge issue for bioterrorism. Let's move on to ringworm. Ringworm is also known as cutaneous mycosis. So that's plural. So mycosis is a disease that is caused by fungi. So ringworm is a fungal infection. The fungi that cause ringworm are collectively known as dermatophytes. Derma for skin, phytes. They look like plants to us, but they are fungi. And we refer to the illnesses that they cause as tinea. So we learned about tinea solium that caused tapeworm, and that was a helminth. They used to think back in the day that this was actually caused by a worm because of the ring shape that it would have, but it is not caused by a worm, but the name is still stuck for the diseases that they cause. These are caused by over 39 different species and all sorts of different genera of fungi, but this is a fungal infection. All right, moving on, the eye diseases now. So,
Eyeball.
huh?
Eyeball.
Yeah, eyeball, exactly. So eyeball, so we have in your eye, basically the conjunctiva, which is our protective membrane that will cover the eye and the eyelids. And that will secrete oil and fluids that will help lubricate the eye. We also have, of course, your eyelashes and things that can protect from things getting into your eye. But your cornea, which is the windshield of the eye, which is actual covering of the eye itself. This is exposed to the environment. It is dome shaped. It's 5 to 6 layers of cells. And basically it's protective for the tissues underneath. And that can become scratched and infected. Then we have your tears as a defense. We have lysozyme in your tears. We have the saltiness of your tears. That can all be antimicrobial and also acts to flush out microbes that could be getting into the eye. The eye is kind of a special situation. It is immune privileged because imagine that you're back in the days of having to live out in the wild and hunt and gather. You relied on your eyes very heavily. And so we couldn't have our immune system causing inflammation that could cause permanent damage to your eyesight. So there is less of immune response in your eye. So you might also be thinking, well, I don't say probably, but you might also be thinking about the central nervous system. Your central nervous system, if you sever your spine, can that ever heal back? No. So your central nervous system cannot regenerate the nerves that damage is caused there. So we can't risk inflammation in your central nervous system either. So your central nervous system is another immune privileged site, but your eye and central nervous system are the two biggies for immune privilege. We can't have inflammation getting out of control in those tissues.
So I've had surgery where they pushed my eye to very gently stitch up the muscles in my eye,
Mhm.
and it took around 13 hours
Yeah.
because they cannot take the eye out of the socket.
Right. And there's a lot of issue with, they don't want to expose it to microbes if they don't have to, because there's not as strong of an immune response in the eye. So we have its own kind of like response going on. The T and B cell response is reduced in the eye. It's regulated so that we don't have as strong there. Our innate immunity is reduced there as well, all to protect the tissues of the eye during the inflammatory response and the immune response. We have most of the chamber itself is cut off from blood supply. We just have like certain vessels that are going into and out of the eye, but that overall chamber is kind of reduced blood flow to reduce microbes from getting to the eye from a blood infection and things like that. So it's hard for your immune system to get there, and that's for a reason, to protect the tissue. So let's talk about some diseases of the eye. And so this is the normal biota of the eye. I won't test you on this, but if you're interested, there is a normal biota in your eye. Microbes and other things like that live naturally in your eye. It has its own biome. It's not sterile. All right, conjunctivitis. Conjunctivitis is an inflammation-itis in the conjunctiva, which is the outer tissues that are protected of the eye. This is pretty common. It can be viral, bacterial, or allergic. And so when we have a bacterial infection, not surprisingly, we've seen how stinky and nasty and goopy our bacteria can get, and they get that way in your own body too. So bacterial infections, we usually have a yellowish or milky discharge associated with that. Yes, this is pink eye. And viral infections, this is going to be more of a clear weeping of the eye. And when you have allergy, you'll have redness of the tissue around the eye just from the allergic response, as well as a lot of like eyes running and nose running and everything like that associated with allergies. So redness and swelling of the eyelid is very common with these infections. And this also can cause photophobia, which I want to be clear, photophobia is not necessarily actually fear of light, but it is used to describe light sensitivity. So the eyes, like the light causing our eyes to hurt more than usual, that's photophobia. So this is pink eye, and pink eye is often caused in adults by poop bacteria getting in your eye, just FYI. I'm not saying that's not what causes it in kids too, it is, but they have a lot of other exposures, yeah. Okay, ocular trachoma, another infection. So trachoma is the #1, and this will be on your test as a recall thing, okay? The #1 leading cause of infectious cause of blindness. So blindness that is caused by infection, this is the leading cause in the world. All right, so there's obviously blindness associated with genetics or trauma or something like that, but blindness is caused by infection. This is the #1 cause in the world. And it's caused by a bacteria called Chlamydia trachomatis. Sound familiar?
Yep.
It should, because we'll talk about Chlamydia trachomatis some more in the sexually transmitted diseases chapter. But Chlamydia trachomatis, the leading cause of infectious blindness in the world, is caused by the bacteria that causes chlamydia, the sexually transmitted disease. It's the same bacteria, but it's not necessarily related. You can still get trachoma from, transfer into the eye from infected genitals. It's the same bacteria, but this is actually more commonly transmitted by flies. And so flies that'll come and land on infected, people and transmit it to other people, or just like touch to touch, eye rub to touch their hand and then you rub your eye and stuff like that. So flies as well as, touch contact. And so this is extremely common in third world countries. We see this as endemic. It's very common to see in Africa, Asia, the Middle East, Latin America, and the Pacific Islands. So this can lead to ongoing and recurrent infections. It leads to this inflammation on the underside of the eyelid that scratches the cornea, and that's what can lead to blindness. You can recover from this. It can potentially cause permanent blindness, but it's actually more common that you can take antibiotics and treat this. All right, keratitis. This is where we have the cornea getting affected. This is usually a more serious infection than conjunctivitis and can lead to actual corneal destruction if it is left unchecked. The most common organisms associated with this are going to be herpes virus 1. So this is like your oral herpes virus, your cold sores, can be transmitted to the eye, can lead to keratitis, as well as acanthamoeba. Acanthamoeba is an amoeba, which is a protozoan. We learned about this in chapter 5. Amoeba are protozoa, and amoebas are found naturally in tap water and fresh water. Tap water, your taps at home, okay? This is why when you do your contact lenses, you're supposed to be washing and rinsing your contact lenses with sterile saline solution, because your tap water has acanthamoeba. So don't just rinse your eyelid or your contact lenses off in your tap water and put them in. I'm sure you've been told that by your ophthalmologist. This is part of the reasons why. This is a potential infective cause of keratitis. I also want to point out acanthamoeba. We're going to talk about it in our neurological diseases in the next chapter. Is also the cause of one of the causes of the brain-eating amoebas. 96% mortality rate. 96 to 97. 96 for this guy. The other guy's 97. So acanthamoeba. When you're washing your hands, be sure you're using soap with your water to get rid of everything. And you're rinsing your contacts off with sterile saline solution. Don't get it in your eye. Right, river blindness. This is a parasitic infection that is transmitted definitely by flies with this guy. So what happens is the fly will land on the eyes of people and transmit this worm. And what happens is that the worm, it can be transmitted by black flies. And they essentially, like the larvae, get transmitted to the people, and then the larvae can migrate into the eye itself, and then the worm lives in the eye, and that leads to river blindness. The worm itself is infected with a bacteria. This is where it gets really meta because the worm can be infected by a bacteria called Wolbachia. Wolbachia is the bacteria. Onchocerca volvus is the worm. So I don't know if you've heard of onchocerciasis, but that's what this is. It's river blindness. Onchocerciasis is the disease. Onchocercus volvulus is the worm, and Onchocerca volvulus can be infected by Wolbachia bacteria. The Wolbachia bacteria are very common in helminths, and most of our infections that are caused by helminths can be controlled by controlling the bacteria that infects them. So this is true with heartworms in animals. So animals that have heartworm infections, we can give them doxycycline to treat the bacteria that's in the heartworms, and it makes the heartworms much easier to kill. And so part of the pathogenicity comes from the worms being infected by the Wolbachia bacteria. But anyways, yeah, you'll get transmitted, the larvae will get into the body, and the microfilariae, which is the tiny version of the worm, will migrate into the eye, and then they live in the eye. Amazing to think about, right? No nightmares or anything. I used to have people stand for my questions, but you guys don't have to do that. I just felt like it helped people start thinking about it more. So which pathogen does coagulase test identify? We're just gonna ask the class Staphylococcus aureus with that one. Which organism causes gas gangrene? Clostridium. This is Clostridium perfringens for this guy. Measles. What is measles caused by? Virus, bacteria, prion, or protozoa? Virus. Excellent. What disease does the variola virus cause? Smallpox. Excellent. And then what pathogen is the most common infectious cause of blindness in the world? Trachoma, chlamydia trachomatis. Yep. Yep. Great job. So before we're going to go live in a little bit, but I want to take some of this back a little bit to start thinking about the application side of the stuff that we just learned. This is our opportunity to start with our critical thinking on these topics that we just covered. So I'm actually going to leave the slide up. I was going to put it in, but I'm going to because we wanted to refer back to anything. So if I tell you, you have a patient coming into your clinic and they have an obvious skin infection, all right? And it looks like they've got some raised pustule, like a raised pustule that is red around it. And it's been like that for a few days. And it's painful, and you can see that it's filled with pus. So we have some things that we could think about, right? Which organisms we think might be causing them and how we might approach the story.
Well, first of all, you want to like put a circle around it, just make sure dermatid necrosis doesn't happen.
To check to see if the redness is spreading, maybe beyond just, we're looking at it at the epidermis level, right? You're seeing it. But what if the infection is spread to the lower levels? Looking for spread of infection in the dermis and the subcutaneous layers. So we could draw a line around it to see if it's spreading. We could do that, and that could give us an idea as well. If it's spreading, what might be more likely, right? So anybody have a guess as to what they think it could be? Staph, probably. Okay, so we'll start with staph, right? So we know, because we learned in the lecture today, that staph, what is it gram positive or gram negative? Gram positive, right? And what shape is the cell?
It's
Coccus. Okay, so we can do a gram stain, right? And what would we expect? What color would the gram stain be? Purple for positive. And we look for the round cells. So if it's Staphylococcus, how would those cells be arranged?
She raised like a chain. No.
Clusters. There it is. So clusters.
I was
So
thinking of
irregular, so that's strepto, right? So if it was strepto, If it was Streptococcus pyogenes, the cause that we see with impetigo, then you'd expect to see the change. They're also gram-positive cocci, but that's a defining factor. So now we can rule out, if we see in our microscope on our gram stain, gram-positive irregular clusters, it's probably not Streptococcus pyogenes. It's probably going to support Staphylococcus aureus, right? So what tests can we do after that with your patient sample to confirm one of the special characteristics of staph? One of the things we talked about today, you know? We could use mannitol salt auger or staph auger, right? So those are good. We could start
We
with
can see what
staph
it
auger to see if it grows on that salty media. We could grow it on mannitol salt
If it
auger.
coagulates.
And we could do coagulates. That's what we were getting at too. So coagulates is very common indicator of cocci, gram-positive cocci that can coagulate. It's almost always going to be staph.
So you use like a blood auger to test that,
We
right?
could do, we would do,
The gelatin thing we did.
it's different. So it's literally just rabbit plasma, like the liquid portion of rabbit blood. I hate to say it, but that is a very commonly used test. We our rabbits don't have to die for it. We can just draw some blood from them and harvest it. But
Very easy.
But the rabbit plasma is what we use. We won't be doing that in our lab just because it's expensive for it. But yes, you would just put your patient sample in the rabbit plasma and see if it coagulates it. If it does, then it's probably got coagulates, which is an indicator of staph. So we could do all of those things, right? So another thing we might be able to do is, other than we know staph is a very common cause of infection in people, right? So we might even be able to, while we're doing these, what we would call phenotypic tests. Those are all phenotypic tests. If it grows on mannitol salt auger, phenotypic. If it is going to react with coagulase, phenotypic. There's no antibody, there's no genetic testing in there, right? And so, right, so we can actually observe the trait of the phenotype, right? So we could do PCR, right? So what does PCR do,
Doesn't it check like the DNA?
right?
It
So
replicates the little segment.
right, it replicates specific segments of DNA. So we can look for specific segments that are associated with Staphylococcus aureus. So in order for PCR to work, what are the 3 steps in PCR? Anybody remember? So I'll start you off, denaturation. First, so how do we denature the DNA strand? What do we do in PCR? Heat, right. So why does heat allow the DNA strands to separate?
It loosens and just like all the proteins.
So it loosens it, but why is it loose? What kind of bonds are in DNA that are loosening?
I want to say like enzyme.
Hydrogen.
Hydrogen bonds. So your nucleic acid bases, your A's to T's and G's to C's, are bound together by hydrogen bonds. You guys starting to see the meta here?
Yes.
Okay, this is why we're doing this, because there's going to be questions that are going to get meta on the final. I'm going to expect you guys to know all of that stuff and apply it all at once. Okay.
Would you normally do a PCR on that? I thought those were kind of expensive.
So yeah, you normally wouldn't. You normally would do just coagulase test or mannitol salt auger and those things, and you could almost certainly diagnose it based on those
Maybe
things.
we need to know.
But
what
this is,
specific-
yeah, so this is just me getting into that other side of it. But yes, you're absolutely right. Most commonly probably wouldn't. It's not expensive to do PCR. It's not cheap. If you don't have to do it, you wouldn't. So yes, we heat our DNA strands. The heat will cause those hydrogen bonds. Those hydrogen bonds are just attractions, right? Between the positive H's and the negative O's. So when we start to get the chemistry of that, with the O's, why are the O's negative?
Because they need.
The oxygens are negative because,
They're
yes.
Yeah.
So,
Mhm.
right, they're gonna hog the electrons, right?
They
They are
want it.
electronegative. So oxygens are hogging the electrons, so they have a negative charge, they're attracted to the hydrogen.
catatonic.
They are going to be positive because their other things are attached to are hogging their electrons. So the positive and negative attraction between the positive hydrogens and the negative molecule on the other side, which is usually oxygen, that's what's holding your DNA together. So the heat actually is weakening that association. So every time you do any cycle of PCR, the first step of PCR cycle is the denaturation. And that's what's happening on the chemical level. So we'll do that, and we'll have primers that we'll add in PCR. That's the anneal step. The primers are going to specifically match up with certain genes that are associated with staph, right? We'll add our nucleotides on there. And then as the nucleotides get added on, like by DNA polymerase, if they're able to add on and build off of that primer that was there to add on to, then that's because you have staph there. It's because you're positive.
And the
Other
third
things
step
won't.
is in that, for PCR, it's,
Third step is elongation.
Elongation, thank you.
All right, so that's PCR. So now your patient comes in, they have a black eschar or whatever it is. All right, they have that black wound on their neck. And what will we assume that would be?
Cutaneous anthrax.
Right, cutaneous anthrax. So what's a test that you could do for that one to check for that?
You can do, let's see, you can do.
Is there any way you could harvest the endospores from it to do like the endospore test?
There you go, yes. So you could culture the wound and then grow it on some sort of nutrient agar or something, and then do an endospore stain in order to see if it has endospores. That would be indicative that it is probably Bacillus anthracis. So endospore forming guy. So if we start thinking about some of the tests that you could be doing there and things that are associated with that. So if I told you that your patient has the measles, what symptoms would you expect they might present with?
Sore throat, dry cough, and you can have the skin lesions, not everybody does, so that you can't like.
Right, the maculopapular rash, or we would have the raised,
Conjunctivitis.
right? Conjunctivitis can be associated with that.
Something like
So just to remind you guys, when we talked about measles can have conjunctivitis as one of the aspects that can develop from having the measles, right?
And just double checking.
So already we need to know about conjunctivitis in order to understand measles and what can be going on with that. What else can we talk about with that stuff? Let's see. We talked about trachoma being caused by chlamydia trachomatis. What type of organism is chlamydia trachomatis? Is it a virus, bacteria, protozoa, or helminth? Chlamydia trachomatis.
It's a virus. No.
Bacteria. So chlamydia trachomatis is a bacteria. Okay. So if I were to ask you, would you use ketoconazole to treat that? No, right, because that's an antifungal, great. So this is what I'm talking about. When you start thinking about chlamydia trachomatis, you don't just think about chlamydia trachomatis, most common cause of infectious blindness. Chlamydia trachomatis also associated with chlamydia.
What would you use for?
You should be thinking about all the stuff that we learned and how it can apply to these things, right? There is a table that I have made for all of Unit 4 for all the diseases and the characteristics that go along with those diseases that is available on Canvas. So if you needed a breakdown of those details, that could be a good like source for the recall stuff. But the stuff of like looking at it as using PCR or using, if I tell you I have an antibody that can recognize this or that, applying that for these things and putting it into practice, that's what I expect you guys to get out of for the final. Yes.
So sorry, so peritonitis, I'm just trying to continue the conversation.
Mhm.
So we have peritonitis in our body. So peritonitis might not affect our skin because we have something to protect
So
against.
so when we're talking about keratitis for the eye, it's more or less that's a term for the cornea
Okay,
in that. So it's an actual inflammation of the corneal tissue itself. I guess.
I was just gonna say on measles, so up until the spot, would it just present like some other sort of like respiratory thing?
Yes, so it'll start out as a respiratory illness. It's not uncommon for us just to have a mild sore throat and a little bit of cough and swollen lymph nodes, kind of just basic cold type stuff.
Another thing was like, I've gotten ringworm from like my cat before, so that means just fungus chilling in his mouth.
So usually it's on their paws or the most common.
you look in their
Yeah.
paws.
He was like a little baby, he was like licking on my skin, I was like, oh, so
So
cute.
it's
And then,
very
like,
common
yeah.
for like cats to have those fungi as part of their biome. And so that could be just not even associated with.
Every year.
Yeah, that was like when we just got him, we hadn't gotten to the vet yet.
Anybody else have any other questions or they want to discuss or look at the big picture stuff?
This is just off topic.
Yeah.
Aren't the worms that kids get in their butts, like the butt worms, aren't those the kind of?
Those are, yes, those are called pinworms.
Yeah.
And those are Enterobius vermicularis.
Yeah.
And we'll talk about those in chapter 23.
I remember learning about those in,
They're very common.
whatchamacallit, high school. I had that animal class, zoology.
Yeah.
I hated that class. When I knew I never wanted to go into animal health care.
Right.
Yeah, I was like,
So I guess one other like kind of question I might ask is, this is not super meta, but to get you guys thinking about the differences between some of these pathogens. If somebody had a smallpox, first of all, we'd be surprised if somebody had a smallpox infection, right?
But
But
who
if
bombed
somebody
it?
had smallpox, so we have agreed that smallpox is caused by a virus. We agreed on that. So smallpox is caused by a virus. So if you tried to grow smallpox on mannitol salt agar, what would happen?
Nothing.
Nothing,
It's-
exactly.
it's
It's a
never
virus. It is
salt.
not a bacteria. It doesn't grow on agar. It has to grow in cells.
Yeah.
In
okay.
cell.
You'd have to like inoculate
Exactly.
it in like a bud or something.
Yeah, so, yeah, well, and we also have human cells that we can grow on like tissue, what we call tissue culture. And so those are, like human cells, like you have probably heard about them in the development of the COVID vaccine, but 293 T cells, which are human embryonic kidney cells. They are not freshly harvested from kidneys. They were harvested like decades and decades ago. And we just regrow them. And then there are, HeLa cells, which are, from Henrietta Lacks. That's literally like what HeLa is Henrietta, H-E,
Which
Lacks,
says
L-A.
God bless her, she didn't get
Right,
paid
God bless
nothing
her,
for
she
it.
got paid
She did not
nothing
get
for it, they
paid.
took her cervical cancer cells, and now it's one of the most commonly used scientific study cells in all of history, and they've just been propagated over decades, so. All right, excellent work, guys, we're gonna head up to the lab and do our lab stuff.